So we're going to now do BLAST to show this and how you can then determine the coding region in your sequence. So BLAST is a tool that we have here at NCBI. It finds regions of local similarity between sequences. The BLAST homepage is listed here. In our earlier Troubleshooting GenBank webinar, we covered BLAST in detail enough to help you. So we won't go through that material again. The URL for the YouTube video, and then the specific time points of that video where we did talk about BLAST. In the previous webinar video, we also listed the help and other NCBI videos about BLAST. So go to that previous webinar to see that information about BLAST. The individual BLAST programs that are useful to you are BLAST-N, BLAST-P, and BLAST-X. When I talked about the common molecular markers that were used, that means we have a lot of data in our databases. And so if you are studying those common molecular marker genes, you're going to get a lot of hits, usually from your sequences when you also do BLAST analysis. However, you might be studying a gene that is not one of the common molecular markers. And then it's important that you consider doing what is called a genomic BLAST, where you will compare your gene sequence to that of a genome for the same organism or for a related organism. And you can then enter the organism name and then see if there's any similarity between your sequence and any genomic sequence data that we have for the same organism or related organisms. So let's go through our first example, and that is 2 beta-tubulin sequences. So we're going to look at the coding region, and then we're also going to look at potential problems with the sequence that is in the data set. The webinar materials, again, are at this shortened link. And if you want to follow along, you want to get this file, that is the demo notes file. The data set that we'll be using is example1 fa.txt. And I've already done the BLAST analyses ahead of time, and these are the 2 RIDs. So I'm going to go to my BLAST results. Again, this is the BLAST page. You can click on the recent results and then enter those BLAST RIDs to pull them up. I already have the first one here in this tab. I increase the font so it's a little bit easier to see. And we have 2 sequences in the data set. The first sequence is the one we're going to look at. It has 693 nucleotides. Again, I'm not going to go through all of the parts of the BLAST analysis. Notice we do have a lot of hits, high scores with the red lines. And notice you can see that these are beta-tubulin matches. I'm going to just collapse the description. And what we want to do is look at the alignment. I'm going to say this now. We said it in the previous Troubleshooting GenBank submissions webinar. Do not ever make a conclusion when you look at BLAST results by only looking at the first alignment. You want to look at many of the alignments to make sure that the BLAST results are telling you a consistent result. The alignment that we have here is the default alignment, and you will see that we have a hit to this GenBank accession number. There are 2 base differences and there are no gaps. And you can see the query sequence starts at nucleotide number 1, and our BLAST database record also starts at nucleotide number 1. And then we go through and you can see the entire alignment. Now, this doesn't tell you anything about the CDS, And that is the trick or the tip that we pointed out in the previous webinar. And that is if you open the Formatting Options section, there is a box called CDS Feature. And then I typically like to see the pairwise alignment with dots for identity so that I can see the differences more easily. So I will change that option there. And then click the blue Reformat button. I'm going to close the formatting options and then just reclose the two sections again so that I can go down to the alignment. And now you can see that there is at least one base difference. But the key here for that CDS feature box being checked is that you can now see the CDS feature that has been annotated on our BLAST database record. You can see that it's beta-tubulin, and the top line is now a translation of your sequence, of the query sequence, using the standard genetic code. So we can see that we have one exon from starting at this GT, because remember, the intron will be ending with AG. And then the tilde symbols indicate the introns. And so this GT is the start of the next intron. So the first exon would be from here to here. Next exon would be from here to here, and so on through the alignment. So your CDS feature would need to include 1, 2, 3, 4, 5 different parts to the CDS feature. And the other alignments in this BLAST result are similar, so I'm confident that that would be the correct answer for my specific sequence. Let's look at the second sequence in the data set. Again, we have good alignments. But now when we look through the alignment, we have a problem here before the last exon. And there's enough of a difference between the sequence in my data set and the database record such that the 8G splice acceptor pair is disrupted, because if this G just before the CTT, if that was included as part of the intron, that would lead to a reading frame shift, and we would get the wrong protein translation for the rest of the sequence. So it's important that you do a BLAST analysis, making sure that your GT-AG pairs are correct and that there aren't problems that would disrupt the reading frame. For this particular situation, you would need to go back to the sequence reads or the sequence traces and perhaps see if there was an error. You might need to resequence the source, the sample again, or you may need to even trim your sequence so that you cut off this last exon, because you cannot get the correct biological translation from the sequence as it is for this last exon. I'm not going to go through the BLAST-X results. They are queued up if you want to ask a question about that, but I think we get enough of our lesson for what we need for our different data sequences in this data set. I'm going to go back to the slides. So a summary for this data set is that the existing annotation for similar records can help you determine the span of each coding region piece. It's confirming the translation using the standard genetic code. And we didn't talk about it in this particular demonstration. It was in the previous Troubleshooting GenBank Submissions webinar, where we went through the gene and CDS strand orientations. And we didn't talk about the complete or partial status, but again, that was in the previous one. But we do have partial for this sequence because we did not have the start codon or the stop codon for the coding region. Let's go to our second example. And this is a gapped sequence. This is actually a human sequence from the HLA-A gene. And it's only exons 2 and 3 that we have data for. We do not have any intronic sequence. Again, the webinar materials are listed there. Let me go back to the data set to show you how the data look. So this is our sequence for example 2. We've defined the information in the FASTA definition line. The first segment is exon 2, and then we have this greater than question 242. I'll explain what that is in a moment. And then we have more sequence, and that is exon 3. When you structure your FASTA sequence with this greater than question and then a number, you are saying that there is a gap between the two sections of sequence that is exactly 242 bases long. So the submitter knows that that intron is 242 bases long. There is another convention that you can indicate a gap when you don't know the length of the gap, and that is to have question mark UNK 100, and that is just a default where we would put a gap of 100 bases, but we're really indicating that we don't know for sure the length of that gap. So I wanted to go through the FASTA file so that you can then understand our BLAST results. And indeed, I pasted the FASTA file exactly as it is with the gap in the BLAST query box, and you can see that the gap is present here in our results. Again, we have good results with the high scores, with the red lines, and we are getting the HLA-A gene hits. And again, we see the alignment, but we have to put on the CDS feature box again. And notice that we have good coding region sequence. However, notice that of the two pieces, that we see the first exon 2 actually, second in our BLAST results. You can actually change that order by sorting by the query start position. And now you can see that nucleotide 1 of our query sequence aligns to the first base of this GenBank accession number. So all of that first part is exon, and all of the second part is also exon. And again, the other BLAST alignments show the same result. Going back to the PowerPoint. So this is a further example about gapped genomic sequences. And this is something that many people have some difficulty with. So if you have a sequence with no gaps, for this particular case, you would have 4 parts to the CDS feature. For this example, where you have 1 gap, you will still have 1 CDS feature with 4 parts. And that is because you have the complete exon for each of the 4 parts. The gap is in the intron. For this part, again, you will have 1 CDS feature with 4 parts. The first part, though, will be indicated as incomplete because it's in the middle of the exon. In fact, all of these are 5' partial, but you're going to start your CDS in the middle of this exon. This is a complete exon. The next exon is complete, and this part is also complete. You're going to have the nucleotide base that corresponds to this point be as part of your CDS feature. For this sequence, you are going to have two CDS features. And the reason why is because you cannot have a CDS feature go across an incomplete gap. And what I mean by incomplete is that you do not have the complete exon that corresponds to this part of the gene, nor do you have the complete exon that corresponds to this part of the gene. So the first CDS feature would have this exon and this little bit of the next exon. The next CDS feature would have this little bit and then the full exon here. So when you have gapped sequences, it depends on where that gap falls as to whether you need 1, 2, or even more CDS features. So the summary for data set 2 is you need to add the indication of the gaps in the FASTA nucleotide file. When you do that, Bankit and Sequin will automatically add the gap features for you. They will automatically calculate the corresponding numbers. If you want more information about how to do that, there is the URL listed on the screen. Exons which are not complete across the gaps must have individual CDS features, but the CDS features can go across the gap if the exons are complete. Let's go to our third data set, and this is 2 messenger RNA transcripts and 1 genomic DNA. And what we want to do is identify the difference between the variants. We also want to identify the CDS and mRNA locations on the genomic DNA. And what we're going to do is to use BLAST2-seq. This is a variation of the BLAST tool. And again, the webinar materials are there listed on your screen. So let's go to our BLAST results. This is the regular BLAST-N for each of the 3 sequences in the data set. Again, we have good hits. And before I scroll down, I'm going to automatically go ahead and change my settings. When you first pull up a pre-computed BLAST result, it does not remember that CDS feature option being checked. So that's why I have to do it each time. So for our first sequence of our data set, we have a complete 5' end. We do have the start codon, and we do have the stop codon. So this would be a complete CDS for this first mRNA sequence of our data set. And the second sequence shows a similar situation. However, if you were observant, you will see that the CDS is slightly longer in the second mRNA sequence. And we'll explain why that is in just a moment. And if we look at our genomic sequence from the data set, we now see different alignment sections. And again, if we do this sort by query start position, we will now see a better biological representation of each part of our genomic sequence. Now, I said that we wanted to determine the difference between the 2 transcript variants, and the way to do that is with BLAST2-seq. So from the BLAST homepage, I'm going to click on Nucleotide BLAST, and then I'm going to check this box, align 2 or more sequences. And what I'm going to do is go to my other window to copy the 2 sequences from our data set. and then go to our BLAST2-seq page. And what we want to do is to see the difference between the two sequences. So I'm going to put the first sequence in the top box and put the second sequence in the bottom query box. So example 3, seq 1 is on the top one, seq 2 is in the bottom one. And when we do a BLAST, we see that seq 1 is missing these bases that are present in the second sequence. And when we look at it in the graphical view, we can actually see that this represents a different exon. We can do that again with a new BLAST2-seq sequence. So this is the second sequence, and now I'm going to do the BLAST against the genomic. And I have that in the separate window. That's the separate file. And you can see that the first exon is 1 to 151. Then there's another exon, third exon, fourth exon, fifth exon. Now I want to point out that notice your nucleotide numbers. Some of them overlap from the messenger RNA. And that is because BLAST does not pay attention to the GT-AG pair. All it's trying to do is align the sequence. So you know that your first exon is going to end between 149 and 151, somewhere in that range. And you actually need to look at the genomic sequence itself in order to see whether the intron starts here or ends here. That is what you need to do to look at the genomic sequence itself. Now, there may be other tools out there that can help you do this. For today's webinar, we're focusing on NCBI tools because that is what we have available to us. I'm going to go back to the PowerPoint slides and just recognize that your nucleotides can overlap alignments, and that tells you that you need to look at things in a little bit more detail. You always want to put the longer sequence in the second query box when you're doing a BLAST2-seq. That maximizes how the BLAST algorithm works. And the important thing that I want you to consider is when you have transcript variants, consider annotating the differences between the variants with the MISC features. And they always require a note of further explanation. Let me show you the feature table that would be appropriate for this particular data set. So our first messenger RNA is 560 nucleotides long. You're going to have a gene feature. The CDS is actually from 65 to 394. And the first sequence is the one without the exon, extra exon. And that location is between nucleotide 148 and 149. And that little caret symbol indicates to the submission tools that that's the location of this exon. And in fact, the note says location of additional exon in variant 2. The second sequence has the extra exon, and we're putting that in as a MISC feed. And then for the genomic sequence, we actually have the gene, and we have the CDS and mRNA that corresponds to the first transcript variant. We have the CDS and the mRNA that corresponds to the second transcript variant. And then we have an additional MISC feature that points out the specific exon that is present in variant 2 but not in variant 1. So this would be an ideal feature table to use for this data set. We've inserted this new slide to correct an omission in the original webinar. If you have a data set that has mixed sequence types, namely mRNA or cDNA, and then also genomic DNA sequences, these must be done as two separate submissions. You cannot have a data file that has mixed sequence types. We did this for the ease of showing the data in the webinar, but I just wanted to make sure that you understood that you need to do these as two separate submissions.